Revolutionary DNA blood tests are transforming how we approach cancer screenings. These innovative liquid biopsy cancer techniques can detect microscopic traces of tumor activity years before traditional methods. At the heart of this breakthrough lies circulating tumor DNA. These are tiny fragments of DNA shed by tumor cells into the bloodstream. Advanced testing methods can identify these fragments as cancer biomarkers, providing early warning signs of disease development. This technological advancement promises to change cancer care forever. Early detection could mean difference between simple treatment and complex battle for survival. This is very interesting. John, tell us more about what's going on here.
Yeah, so this is really interesting. So they've shown that cancer leaves genetic traces in the blood long before traditional diagnostics would spot a tumor, which is crazy. And it does make sense because cancer grows, right? It starts at one point and just grows. So they used blood samples, which were collected decades ago from participants in a long-term health study. They analyzed cases where individuals later developed cancer. With this new cutting-edge genome sequencing, so again, we couldn't have done it with old technology. We need new technology. They were able to identify specific DNA mutations, which they termed circulating tumor DNA or ctDNA. And they were able to detect it sometimes more than 3 years before any clinical diagnosis, which is awesome. I think cancer is something that I feel could have been solved already, and it's just been delayed. And this is great. This is going to help with detection so that pharma can keep making their money on the drugs, but this is going to help with people who can potentially a better chance of survival. So the thing with this is they faced a sensitivity challenge because newly formed tumors are tiny, and they only released a minuscule amount of what they're calling ctDNA, which made the detection extremely difficult. So think about it looking for a needle in a haystack, right? So by using these advanced sequencing techniques, 1 out of 26 individuals who were later diagnosed with cancer, the ctDNA was detected in eight of them at the time of the blood sampling. And 4 had cancer DNA signatures in their blood more than 3 years before diagnosis. This is crazy. Again, we're using newer technology though, right? So which makes sense for an advancement like this. What are your thoughts?
This is very cool. So this does whole genomic sequencing, revealing cancer mutations at extremely low concentrations, like you mentioned, in preserved blood samples. Fragmentomics use machine learning to decode specific DNA fragmentation patterns for early detection. The multimodal liquid biopsy improves accuracy by combining sequencing and the fragmentomics for stronger detection. The circular tumor DNA that you mentioned, the ctDNA, serves as a reliable biomarker for multi-cancer early screenings and monitoring. Current tests show 87.4% sensitivity and a 97.8 specificity, nearing clinical viability standards. This also means a single blood draw could screen for multiple cancers rather than right now you need to have one for each screening test. It's interesting that machine learning is behind a lot of this to help identify and detect subtle DNA patterns. So this is really getting down to the future of personalized medicine.
Yeah, it is. And there was another article, we won't talk about it. They were using mRNA to code your cells to attack cancer cells. So that's something different. But there's just speaking to advancements in cancer treatment, right? The one caveat here is that not all cancers shed detectable amounts of ctDNA at the same rate. So that could limit the effectiveness of the test for certain types of cancer. So the results from this case study was highly controlled and may not fully translate to population-wide screening. So obviously there's going to have to be ongoing large-scale trials to assess this impact, but the types of cancer that the study included were breast, colon, liver, lung, pancreas, and rectal, which shed different amounts of ctDNA. So I love this. I love where they're going with this. This seems like it's a viable detection methodology based on what I read. But again, I'm not an expert. I'm not an oncologist. So what do you think, Craig?
Yeah, I love this because, one thing maybe we touched on, but didn't really, early cancer detection increases survival chances dramatically and minimizes the treatment complexity and invasiveness. So the earlier you can detect it, the better your odds of beating it and the less drastic the treatment has to be. Yeah. So if we can now detect even 80% of cancers 3 years beforehand, that means significant quality of life for people who are suffering from this, from cancer. They get it detected, and hopefully get treatment that's much lighter and easier and get it resolved within a very short period of time, rather than, the extensive chemo and stuff that people have when your testing comes too late. So I love this. I understand that it's a caveat. I understand also that this will probably be expensive. So it's going to take a while to get the price down, because we are talking about personalized medicine. It's your DNA. We're scanning for defects and stuff, and it's specific to you. So there's things that have to happen there. But I love where this is heading. Every time we see when they apply AI to medicine or to science and whatnot, we're starting to see major breakthroughs, and they're coming faster and faster, and it's great to see.
When they apply it properly with people that know what they're doing, as opposed to just trying to use an AI and getting hallucinations and crap. So,
we're not talking about somebody typing into GTP writing a school paper. We're talking about finding new proteins, comparing, finding medicines that don't interact negatively to each other, all and this kind of stuff where it has to be used properly. The clinical testing has to be done, but it's interesting to see that we are making gigantic leaps forward in science as a result of this.
Yeah, this is cool. Yeah.
So we'll have to see, how long is this going to take till we're going to see it live? Do you know?
No clue. I'm guessing maybe 3 to 5 years.
Yeah, I'm looking through my notes here. I didn't see anything in here. No, I didn't make any notes on the date, so I guess I didn't see anything in the article. So I'd love to see this. We all know people who have had cancer and affected this in our life, and it would be great to see if, as a society, we can get it and treat this early, and maybe not put an end to it, but significantly reduce the amount of people who have to go into long-term care or deep chemo as a result of getting cancer. So let's hope this progresses fast.
Yeah.
We'd love your feedback on this new cancer detection, and I'd love to hear your thoughts. Leave your comments below. What do you got going on?
Not much, just working away, helping clients get government funding, and working on my startup, Lighten.community. If you want to sign up for the waitlist, go to Lighten.community. But if you need help with government funding, find me on LinkedIn. How about you, Craig?
Well, depending where this goes out, August 19th is our next Flowgrammer event, and then September 16th, where we do business automation, get the Toronto builders together, and we have some great discussions. We were mentioning at the top of this that there were some great discussions around Claude versus some of the other platforms, and there was some great discussion about how they're all going to go away, Lovable and Cursor, because they're burning billions of dollars, and two, the actual AI companies don't want to lose that revenue to these third-party companies. So OpenAI, Claude themselves want that revenue. So it's only a matter of years before that is built into these products. So it'll be interesting to see. But check everything else at torontostarts.com or flowgrammer.ca for everything we say and do. And awesome. John, if they're looking for non-dilutive funding, where do they go?
Yeah, find me on LinkedIn, John Irwin.
Thanks everyone for watching or listening, and we'll talk soon.